So in cellular respiration, there's two kinds. We have anaerobic and aerobic respiration. So in this video, we will be finishing up our aerobic respiration, which is what occurs when there's oxygen. In a previous video, we looked at glycolysis, the oxidation of pyruvate, and the Krebs cycle. And so here, we'll be finishing off with the electron transport chain. And then following this, we'll look at fermentation and what happens when conditions are anaerobic. So, here we go. Oh, I want to point out, though, is that glycolysis happens in the cytoplasm of all cells. So glycolysis happens first, whether it's aerobic or anaerobic conditions. We're going to split sugar, and that's our first step. And then if there's oxygen, the mitochondria will be utilized. And if there's not oxygen, the fermentation will stay in the cytoplasm of the cell. Okay. So here we are in the mitochondria. This is the matrix where the Krebs cycle happened and the oxidation of pyruvate. So you have a lot of electron carriers have been made or reduced in this matrix area already. So it's kind of helpful that the electron transport chain is located here. Now these things are called cristae. These are the folds in the inner membrane. And the reason that it's highly folded would be to increase surface area, to have to maximize the space possible to have electron transport chains. Okay. All right. So, let's go ahead and now zoom in to the ETC. So here we have the intermembrane space, which is this area that we just saw. This is where the protons are going to accumulate in the intermembrane space. So here we have our inner membrane and our matrix. This here is the electron transport chain made of enzymes. And when we talk throughout the year about cytochrome C is one of these enzymes involved in the electron transport chain. And so just to point it out to you now so it kind of sticks in your head a little bit. All right, so here we have a reduced electron carrier that in the previous steps, glycolysis, the oxidation of pyruvate, and the citric acid cycle, where we oxidized our food and we broke bonds, we took the electrons and we reduced electron carriers. So now here we have our electron carriers are going to be becoming oxidized. So our electron carriers are going to donate their electrons into the electron transport chain. So what we see here is that look at that oxygen. So here we have oxygen, and oxygen is very electronegative. It attracts the electrons. And so in the mitochondria, aerobic respiration, our use of oxygen is the final electron acceptor. It's going to be pulling the electrons down the electron transport chain, attracting those electrons. And then as the electrons are traveling down the electron transport chain, protons are pumped into that intermembrane space. So when the electrons go from one protein to the next, they're giving up a little bit of their energy to pump that proton into that intermembrane space. Now here we see we have that reduced electron carrier. It's now back in the NAD+ form. It can go over to the Krebs cycle to be reduced again. It can go to the oxidation of pyruvate and be reduced again, or it can be used in glycolysis and be reduced again. So here we have basically recycled our electron carrier. It showed up as a reduced electron carrier. So here you have your reduced electron carrier with the hydrogens and the electrons. And as they become oxidized, the electrons enter into the electron transport chain, pumping protons up into that intermembrane space. And so this electron carrier right here is now available to be reduced again in the other steps. We've recycled it. Now you also notice here that the oxygen has gained those electrons, which we'll see what happens with that in a few seconds. But here we have all of these protons are going to be accumulating in this intermembrane space. So we have these protons, and what we've done is we've established a proton gradient. So we've created this proton gradient in the intermembrane space. And obviously positive ions aren't going to voluntarily go towards other positive ions. And so this is why we use the word pump, that it's pumping and forcing them into an area where they wouldn't naturally go. And so we've established a difference in charge across the membrane, all because we oxidized these electron carriers. So here, the electron carriers are the ones that have brought the electrons and the protons to the electron transport chain, and as the electrons travel down and oxygen is attracting them, pulling those electrons down the electron transport chain, the protons are being pumped up into this intermembrane space, creating a proton gradient, a difference in charge across that membrane. All right, so as we keep on going, now logically, let's think about what we've seen in aerobic respiration so far. When a molecule gains electrons, it also gains a hydrogen. So when we look at this, we're thinking, well, the hydrogens are all stuck up here. How are they going to get through, right? And so we have a protein called ATP synthase. So ATP synthase is where the protons will flow through and make ATP. So protons are going to flow through ATP synthase. And when they flow through ATP synthase turns and does phosphorylation. ATP synthase will add a phosphate, an inorganic phosphate, which is P with an I, an inorganic phosphate to an ADP, doing phosphorylation. And so ATP is made when protons that we forced up into this area, into the intramembrane space, and now they're going to flow basically by diffusion. They flow from an area of high concentration to an area of low concentration, down their electrochemical gradient, high concentration of ions and positive charges. They're going to try and they'll repel each other, cause them to flow through ATP synthase, which turns ATP synthase and does phosphorylation. Bam! Okay, but now the cool thing is we have these hydrogens back into the matrix, where here these hydrogens are going to be attracted to that very electronegative oxygen that's gained additional electrons, and this is where water is formed in aerobic respiration. So really we can say that this oxygen here was gained electrons, gained hydrogens, so our oxygen is reduced to form water. And so you saw again some more ATP were made as hydrogen ions were flowing through ATP synthase. Okay, so water is formed by oxygen gaining electrons, gaining hydrogens. So basically oxygen is reduced, forming water. Okay, so when we look at this here, this proton gradient, what we see, we have these protons flowing through ATP synthase. This is called chemiosmosis, the diffusion of ions from high concentration to low concentration. And basically, this is from Google Images or Google, more scientific for you. But what's happening here is it specifically relates to the making of ATP by the movement of hydrogen ions across a membrane during both the light reaction in photosynthesis as well as the electron transport chain here in cellular respiration. Okay, so when we look at this chemiosmosis, here we have them flowing, and because the ATP synthase turns when they flow and makes ATP, these, you're going to make an incredible amount of of ATP from each glucose. We get about 30 to 36 ATP for each molecule of glucose. And we're going to get a whole lot of them here. Now, earlier in aerobic respiration, in glycolysis and the Krebs cycle, ATP was made by substrate level phosphorylation. Here, we took that inorganic phosphate. In this one, we are not getting phosphate from a substrate. So, this is not substrate level phosphorylation. If you remember at the beginning of the electron transport chain, we oxidized this electron carrier. So here we oxidize the electron carrier, and that electron carrier being oxidized is what established this proton gradient. The proton gradient exists because we oxidized electron carriers. So as we oxidized electron carriers and the electrons travel down and the protons got pumped up, we create this proton gradient, and now we have them flowing through by chemiosmosis, making ATP. So we call this process, if you combine both of those things, we have oxidation and we have phosphorylation. We call this oxidative phosphorylation. So this type of making ATP is called oxidative phosphorylation because we oxidize the electron carrier and phosphorylated ADP into ATP. So oxidative phosphorylation. And this is where the majority of the ATP is made in aerobic respiration. Okay, so when we look at a picture here, this section is the electron transport chain, chemiosmosis, together, oxidative phosphorylation. All right. So when we look at respiration, we see that our food molecule is oxidized, leaving us with carbon dioxide. And then our oxygen gets reduced, forming water. Awesome. So in aerobic respiration, though, we need food, oxygen, but we also need electron carriers. So we got to think, well, what happens if we run out of NAD+? Right? Can we do aerobic respiration? Or any respiration? Can we do glycolysis if we run out of NAD+? And if we think about it, the majority of our electron carriers are going to be oxidized at the beginning of the electron transport chain. So here, this NAD+ can be used in glycolysis, it can be used in the Krebs cycle, it can be used in the oxidation of pyruvate. But if we're not in aerobic conditions and it's anaerobic, then there is no electron transport chain. So if there's no electron transport chain in anaerobic conditions, how do we get NADH oxidized back to NAD+ so we can continue glycolysis, right? So that's where where fermentation comes into play. So here we have aerobic respiration, but in conditions where there isn't oxygen available, we're left with anaerobic conditions. So in glycolysis, we are desperately trying to make ATP. If there's no electron transport chain, that's our only source of ATP is glycolysis. So in order to keep that going, then we're going to do fermentation. Fermentation will be able to allow glycolysis to continue. And we can at least still make we can still make ATP. All right. So in glycolysis, let's remember, at the end of glycolysis, we made pyruvates and electron carriers. And if our main concern right now in anaerobic conditions is to make ATP, then these are just wastes at this point as waste products. So here in this step of glycolysis, we need NAD+ to be reduced. If you run out of NAD+, you're not going to be able to make your ATP during glycolysis. So in an animal cell, these two things were wastes. We only are concerned about the ATP during anaerobic conditions. That's our main source of how we're getting ATP. So what's going to happen is we need a place to turn this NADH back into NAD+ so we can continue glycolysis. Well, pyruvate's not being used for anything. So in a chemical reaction that happens, we're going to oxidize our electron carrier and basically reduce pyruvate. And when that happens, pyruvate will be converted into lactic acid. And this is going to reduce our electron carriers so they can be used again to continue glycolysis. So if I look here at this slide, here's for people who like to visualize it. If during anaerobic conditions, my main concern is this ATP, and I need to keep glycolysis happening to make ATP, well, I got to make sure I don't run out of NAD+. So after glycolysis, I have two reduced electron carriers and a pyruvate. So what's going to happen is I'm going to need to oxidize this electron carrier. And when I do that, pyruvate is converted into lactic acid or lactate. And now this NAD+ can be used again in glycolysis. So in anaerobic conditions, fermentation will follow in order to recycle or regenerate NAD+ and allow glycolysis to keep on happening. If we did not have this part, then our NAD+ would run out and the glycolysis would eventually stop and we wouldn't get any ATP. Now in bacteria or yeast, the reaction and the idea is the same. Well, the reaction's not the same, but the idea is the same, except for the products in alcoholic fermentation are going to be a 2-carbon alcohol called ethanol and a carbon dioxide. But again, it's to oxidize the electron carrier to continue glycolysis.